Okay, so we are going to look at tools which are routinely used for understanding or studying the developmental biology, or which are used in developmental biology. So for example, one of the fundamental question we work on is to identify cells of different fates. For example, we are to differentiate between muscle and neuron. So an obvious and first step is we can look at morphology. A muscle cell looks very different because it's a multinucleated, if it is a muscle fiber, or if it is a mononuclear single cell, it looks like this. And a neuron, its morphology is different with dendrites and then the axon, etc. And we can do some histological stainings to see, and these histological stainings will make the morphology more clear. And we can correlate, yeah, this morphology is of a muscle cell and this is a neuron. However, in 21st century, while studying developmental biology, we focus on molecular markers. We have to use some cell type specific markers, and that can be a protein with specific enzymatic activity. If we have an antibody against that protein, we can simply do immunostaining of these two cell types and then see if that particular neuronal marker, muscular marker is expressing in those cell types we are working on. If we don't have protein-based tools, like if we don't have antibody, we then can still use messenger RNA for tissue-specific genes or cell type-specific genes. And for that purpose, we use in situ hybridization, or we can simply isolate total RNA from the two cell types. Let's say we have cell type A, cell type B. It can be a totally relevant question, like we have wild type cell, a normal cell, and a cancer cell. So it's a lung cancer. So lung cancer, normal. And then we can use, in such a case, we'll use some oncogenic marker which is expressed in lung cancer or neuroblastoma, glioblastoma, etc., whatever cell types we are working with. And if we don't have the antibodies, we can do in situ hybridization. That particular gene will be expressed in messenger RNA, will be expressed in neuron, absent in muscles, or expressed in, let's say, cancer and off in wild type. We can also do RT-PCR. We can isolate total RNA, and instead of in situ, we can do reverse transcription PCR on the cDNA made from these different kinds of cells. Also, we can use a reporter gene. That is a altogether different strategy. And I'll talk about reporter gene later on. But remember this, developing a tissue-specific reporter or reporter gene which you can use, it can be a GFP tagged, luciferase tagged, or a tissue-specific promoter driving GFP, tissue-specific promoter driving luciferase. So this is a very useful tool, but we'll talk about this in detail in later part of today's recitation or maybe next week. So if we have to see, so this was about trying to identify cell fate. If we have to, see if we can change cell fate A to cell fate B, if we can, if we want to work on, changing cell fates. Again, we can do some conventional tools like transplantation, which we call cut and paste, etc. Tools. And in cut and paste, you basically play with the time and the space. So for example, you use embryo, and this is an early stage embryo. You take a presumptive, let's say mesoderm, and you graft it to uh, to a late stage embryo, and then try to see if this early stage cells which are not yet committed, if they develop to the same cell fate, or in the new environment, the late stage cells, all of them are already determined. Can they convert them to their fate, or it makes what it was supposed to make here? So you do such cut and paste experiments. You play with both time, early, late, or late can be also transplanted in the early, and space as well. For example, let's say we take same stage embryo. We take, you remember the experiment from Hans Beeman. So you take a piece from dorsal lip of blastopore, and instead of transplanting here, you transplant it at a different location. So the location of the cell is different, and then you see if it develops into same thing, or, and what we learned in that case was that it developed in a whole small sized embryo. So such cut and paste experiments or transplantation experiments, you maneuver both time and space. Similarly, another experimental strategy we do is we simply take out the cells from its natural environment. And taking it out means, let's say we have this embryo, we take these cells, you place them in tissue culture in a artificial environment. Now, what have you done? You have actually eliminated all the cellular environment, the cell-to-cell interactions in its natural context, and then see if these cells still behave, still develop into same fate had you left them in the original position. And what you find, whether in such kind of experiments, you find whether cells are already specialized, if their fates have been specified or no. So what you, I'm sorry, I had to switch it off. Yeah, here it is. So then what we do, we also do experiments in which we ablate using radiations. You can use UV radiation, X-rays, or laser ablation is very common. For example, let's say you have a tissue, and let's say I draw fruit flies wing imaginal disk. And these are the cells which are going to make adult wing, okay? With this laser, because laser is very targeted, you can target single cell maybe, or just few cells dia. You kill cells here, you pointedly kill cell, and then you can study the behavior of the neighboring cells and the developmental morphology of the wing which develops from this wing, okay? You can also do UV mediated ablation. So UV simply can cross-link many of the cytoplasmic factors. You know, it can not only cross-link. You may have heard about thymine dimers, the thymine in the DNA, but it can cross-link messenger RNA proteins in the cytoplasm as well. And you, by such an action, what you have done, you have basically depleted cell from some of the factors. Or you can use X-rays and you can ablate the very rapidly dividing cells by exposing to X-ray radiation. You can block those cell divisions as well. And other strategies of uh, ablation is you can use drugs or toxins, for example diphtheria and ricin. These are lethal toxins. Ricin is also used as bioweapon. It's such a toxic chemical. So why you so all these stuff, very simple tool, cut and paste, to isolating cells out of its natural environment, or ablation. You are basically trying to see development of cells into a specific fate, and whether by maneuvering with the environment or space and time of these cells which are being uh, manipulated using these tools, all these three strategies, whether you can convert one kind of cell to another kind of cell. Now, more targeted approaches, which are more relevant for our course and in the labs, in cell and molecular biology lab, these are genetic tools or gene-based approaches, or I call them molecular tools. Uh, for example, naturally, when cells lose their fate, uh, that can be due to genetic mutation, and genetic mutation normally uh, spontaneous mutations are like 10 to power minus 6 or so. Just a hold on, I'm getting an emergency call. Ji, Assalamu alaikum. Ji, wa alaikum salam. Okay, so apologies for this slight disruption. So the genetic mutations are normally when genetic mutations occur, one of the lethal consequences which can happen naturally is a normal cell fate. A cell loses its memory, it loses its cell fate, and cell goes from normal to a disease state due to due to a specific mutation in these somatic cells. However, uh, you can also, when you want to study developmental biology, when you want to understand cellular function in the context of development, you can do genetic mutations, and you can do very precise uh, genetic mutations using homologous recombination. So you know that a gene X is important gene, and you want to study its function. So you can use homologous recombination, develop knockout mouse, or nowadays using CRISPR, you can simply knock out the specific gene you're interested in. Or conventionally, people had been using EMS, MMS. These are the different, or even X-ray radiations to create mutants. Okay. Also, you can so specifically inactivate gene function. Like we said, you have a gene X, and you specifically already hypothesize, you have an educated guess that this particular gene is important in specific cell fate or specific developmental function, you will specifically knock it down. So here I use knock out. Knock out means if this was the normal gene in genomic locus, in your knockout, this gene is gone, delta, deletion. Okay. Deletion means, or your knockout strategy has either you can kill its open reading frame or altogether the genetic locus, the gene is removed, excised out. The other strategy is knock down. You can use, double-stranded RNA against your gene X. You all have heard about RNAi pathway. So siRNAs against your gene X, and in that particular organism or cells you are playing with, again, you can play with specific time, cells, spatiotemporal regulation during development, and then see if, since siRNA is going to go and recognize homologous messenger RNA, and dicer then cleaves this messenger RNA, which is being transcribed from wild type locus. Now, in this case, we call this knockdown because the expression of messenger RNA is being diminished. Transcription is taking place from the gene, but messenger RNA is being targeted, and it's never 100%. So the protein concentration is going down. While in case of knockout, you have removed the gene altogether. Then you can also use ectopic expression. Let's say you have found that gene X is, let's say here, again, we can bring in fly imaginal disk. Yeah, Momina?
सर आपने जो कहा है कि हम जीन लोकस को रिमूव कर सकते हैं, will it not come under genetic mutation rather than specific inactivation of gene function, क्योंकि हम तो उसको पूरी की पूरी जीन उड़ा रहे हैं.
So this is specifically inactivating gene because you're targeting a specific gene. In a way, this is also a genetic mutation. You are creating a mutation. You are creating a null mutant. But when we use large-scale word about genetic mutations we create by using mutagenesis process like EMS, MMS, or in case of plants, using tDNA insertional mutagenesis, in fruit flies using P element mediated mutagenesis, etc. But this homologous recombination, not only the knockout not only comes under inducing genetic mutation, but also about specifically inactivating a gene function.
Okay, sir.
Clear?
Yes, sir.
So let's now assume that the gene X we are working on, we hypothesize that it is involved in expression of gene X is specifically involved in wing formation in fruit fly. Okay. Now the point is, what we will do, we say, okay, we take gene X, we clone it in such a way, and we will use the tools that now instead of wing, we express this gene in the halter. So now gene X is being expressed away from its natural location, and that is what is referred to as ectopic expression. So now when X is expressing in halter away from its natural location, we are going to see if halter becomes like a halter, like this, or halter becomes a wing as well. May not be a perfect wing, but becomes like a wing. Or we talked about PAX6. You remember we talked about PAX6, which is a master regulator of in the eye development. So what they did, they used US sequences, upstream activating sequences, cloned fruit fly eyeless gene, and made transgenic flies. Now, these transgenic flies were used to cross with tissue-specific GAL4 driver. For example, wing-specific GAL4. Now, ILS, which is normally expressed in, normally ILS is expressed only in the eye imaginal disc. But now you are inducing into wing, and then you can see if on the wing I see red eyes or not. Okay? And these are the photographs you have already seen. Now, so I went through these cut and paste. So this is basically what cut and paste is. I explained you there as well, but still. So you can play with space on the same stage embryo or a different stage embryo, and then try to see how the cell fate develops. Also, while doing these kinds of experiments, we can also mark cells. And marking cells means you early on during embryo, for example, you mark cells here. Let's say these cells become marked by GFP. Okay. Now we know that there are different imaginal discs. I just showed you eye, wing, halter, then the brain imaginal disc. I'm talking about fruit flies. Or you can think of mouse or any other organism. Now this embryo, when grow, these glowing cells, if they become part of brain and never in halter and wing, etc., you say, okay, these cells are basically part of the future brain. So from here all the way to brain disc and to adult fruit fly head, you will see GFP. So the whole cell lineage, all the cell lineage, here few cells are there, but then they went through morphogenesis to the organogenesis, differentiation stage, and then you see adult tissues, and you can follow the cell lineage. In fruit fly, these are very specific tools. We call them Markham clones. Okay, I'm not going into detail of this, but I just brought this at the front so that you can understand what I'm going to talk about. Now, also people could use vital stains in the past. So these stains or dyes, they can be injected, but problem with these dyes were as embryo grows, they get diluted, and you can only trace them until certain stage. And they have also used radioisotopes like radiothymidine, and they could label only mitotic cells where DNA replication is taking place. They could see existence of this isotope. Also, in last 15-20 years, we saw caged fluorescence as well. These are fluorescent compounds, fluorophores, which you inject into the cells, and then using a specific laser of specific wavelength, when you hit the cell, these cage compounds start emitting fluorescence. Okay, and so these caged compounds, as cells grow, they keep within the cells, and at specific, when you want to do analysis, you hit with the laser, and if your compounds have been through specific cell lineage, all that will lineage will glow. Molecular markers, as I said, we in our labs in cell and molecular biology lab, people who are interested in molecular basis of development and want to understand genetic and molecular basis of development, they use molecular tools like, antibody, I already explained you, tissue specific markers. Tissue specific markers are, for example, mouse, coat color, Drosophila, breast cells. We in my earlier lectures, I told you, for example, on the Drosophila thorax, we have a very specific pattern of breast cells on the male first pair of leg. You have the sex comb structures, the fruit fly ventral side of abdomen as well as dorsal side of abdomen. There are very specific pattern of bristles present, which will differentiate one body segment from another one. And you, by following these markers, you can very easily, cell-based markers, you can see the cell phase. Now, there are non-tissue specific markers as well. There is a very famous experiment, we will learn about this, where they can take chick neural crest cells, transplant them into quail or vice versa. And they want to see whether in the quail, the chick neural cells, they contribute to quail neural tube formation or no. Then of course nucleolus, etc. So there are many cell-based markers. Also, you can use transgenic markers. You can use LAGZ, GFP, luciferase, etc. You can use, for example, as we were just talking about, you can use ILS promoter and clone GFP. Now, your flies, their eyes will become GFP positive, because this promoter will only be active in eye tissues. Or you can use, let's say, wing-specific enhancer and drive expression of your LAGZ or GFP or whatever. And once you will see, you can very clearly see expression of LAGZ or GFP, etc. And that will help you understand where in the cells wingless is active or is supposed to be active. So we stop here, and if there are questions, let me know, because we'll go slow with these tools. I would like you to assimilate. I would like to go back and try to think about using these tools, whatever we have talked about today, and design an experiment for yourself. Rather, define a question and then think of answering that question using some of these tools we have covered so far. G, any questions? G, Fariha?
Assalamu alaikum. So, sir, what I understood from what you discussed about the knockout and the knockdown. So you mentioned that knocking out means it can be either deleting the entire gene locus or, yeah, that's what you said. Okay. So the next thing that you mentioned was knocking down, and you mentioned that if we are using the siRNA and knocking down the expression by targeting the messenger RNA. Now, my question is that knocking out, basically, what I understand is that it means knocking out the product of the that gene, that is the protein, right? So if we are not supposed to get the protein as a result of that entire transcription and translation, but what I understood from your your definition, if it's basically the entire open reading frame of that is to be removed, like the locus is to be removed. But if what will happen if we just disrupt the reading frame of that gene, that protein, and then it will not be translated. So that can also be referred to as knocking down the expression, right?
So I think I need to explain again. So make one thing clear. Knockout means knockout, it means if this is a gene from here to here in the wild type, your essential target is basically getting rid of the gene product, which is the protein. You cannot get rid of protein if gene is intact. So what you do using homologous recombination, you design a strategy in which you can take from here to here, either the entire gene can be excised, or you can simply, as small as possible, you can simply remove the initial few codons, ATG to something, so that even the messenger RNA will be made. There is no reading frame there. It is not necessary you remove from here to here everything. Once you remove from here to here, this is the safest. There will be no codons present in the locus. So that's why people want to remove everything. But even if you remove just few nucleotides, that can work equally good as from here to here, because RNA pol 2 will go through this, but messenger RNA, which will be transcribed, will not have the start codon for ribosome to translate, so you will be missing the protein. But mostly knockout is like you remove total gene. Okay.
Okay, thank you.
Then the second thing you said was the knocking down. Okay. Knocking down means the gene is intact in the cell, its messenger RNA is being made, it's being transcribed, and then protein is being made. What you do in this cell, you add double-stranded RNA, which is homologous to this messenger RNA. It will recognize, bind to these messenger RNAs, and all of them, due to RNAi pathway, will get degraded. Very few messenger RNAs, still wild type, will be intact, and small amount of protein will be made. So this is knockdown. Did I answer your question?
Yes, sir. Thank you.
Any other question?
Sir, I wanted to ask the same question again. Sir,
Yeah.
मैं confused हो रही हूँ because आपने genetic mutation अलग लिखवाया and then specific inactivation of gene function अलग लिखवाया है. लेकिन ये जो आपने knockout बताया है, ये एक तरह से genetic mutation की category में ही lie करेगा ना? Because हम gene को eliminate कर रहे हैं, right?
जी, बिल्कुल.
Okay, sir, that's all I wanted to ask.
Any other question?
So, sir, so one thing I'm wondering right now is that in the knockdown wala pathway, the gene is intact, but the protein it's getting diminished. The mRNA is getting degraded, and the protein ki jo expression is going down. So, is there a way that the cell realizes that this protein of interest is no longer being expressed at the right level, and if it's involved in some very specific, very important, very vital function, and the cell says okay, this vital protein is now not being expressed, and somehow the original transcription goes up?
Whose transcription goes up?
जिसकी हम knockdown कर रहे हैं.
Why its transcription should go up?
So I'm just wondering if the cell can somehow realize that this was the protein that I required.
There can be such a mechanism. Why not? I mean, there can be such mechanism where there can be some feedback loop where cells control their protein concentrations. There's auto feedback loop where protein product of a gene tells its own gene that my concentration is enough, you don't need to transcribe more. And an alternate such a hypothesis is true that when such an important protein is going down, You know, there can be a message to the DNA locus to transcribe more, but you have double-strand RNA, they will be still not able to rescue the protein because the messenger RNA will be degraded. Did I answer your question?
Okay, so thank you.
Any other question? No more question. So I heard that there was a student who said she or he has 15 minutes overlap, I think. I don't know who he or she.
Yes, sir, I do.
Who has this? Who is the student?
Sir, Shirjeel.
Shajil, why you did not tell us when you were finalizing all this recitation?
Sir, actually, I wasn't in the class today.
Yeah, but we finalized last time in the last class. So you were not there in the last class either.
No, sir, I was in the last class. I guess it was finalized today.
No, it was finalized in the last class that we will have Thursday, 6 to 7. So it's no problem. We can start from 6:15. So your class finishes at 6:15.
Yes, sir.
So we can go from 6:15 onwards. Anyone else has any objection? Okay. So I'll also upload papers about the lectures we just started today. But first we will go through these couple of recitations about tools so that when you read those papers, you know, it may help you. And also we'll make a little progress in our lectures as well. But you should start reading those papers. All 4 papers I'll upload. You should start reading all 4 papers. Read them again and again and again and again, unless you really understand everything. Okay? If there are no more questions, thank you very much. Allah Hafiz.